Spherical nucleic acids, or SNAs, consist of densely packed, highly oriented nucleic acids typically arranged on the surface of a nanoparticle. Most linear and circular forms of nucleic acids derive their structural form from the hybridized duplex. SNAs, however, derive their structural form from an inorganic nanoparticle core, which acts as a scaffold for covalent binding of single or double-stranded nucleic acids in a radial array. In the same way that fullerenes are a unique form of carbon, SNAs are a third important form of nucleic acids, defined and differentiated in terms of structure and function. The three-dimensional architecture of SNAs imparts upon them interesting chemical and physical properties, making them superior to linear forms of nucleic acids in many biomedical applications, including gene regulation. In order to cross the cell membrane, negatively charged linear nucleic acids must be pre-complexed with carriers like lipoplexes, peptides, or viruses. SNAs do not require such carriers and enter cells in high quantities despite their negative charge. Their high density of nucleic acids leads to interactions with membrane-bound scavenger proteins. As a result, they are taken up through endocytosis as single-entity agents, eliminating the need for ancillary transfection agents, which often cause harmful side effects. Inside the cell, SNAs are shielded from degradation and do not trigger an immune response. In comparison, linear forms of the nucleic acids of the same sequence are rapidly degraded and are highly immunogenic. Once the nucleic acids reach the target transcript, both linear and spherical nucleic acids can utilize either the antisense or RNAi pathways to regulate gene expression. In the case of the antisense pathway, SNAs have binding coefficients that are 100 times higher for complementary sequences and bring about a more efficacious and persistent knockdown than their linear duplex cousins. All of these unique properties stem from the densely packed spherical arrangements of nucleic acids that define the SNA. One can even dissolve the nanoparticle scaffold to create a coreless or hollow form of SNA, which exhibits many of the hallmark properties of the original nanoparticle conjugate. SNAs are revolutionizing the field of biomedicine by enabling powerful new applications in medical diagnostics and therapeutics that can be used to analyze or treat many diseases with a genetic basis.